Matt, we're going to wait on your okay to go.
Yeah, I'm going to paste the new YouTube. So we're live right now on YouTube.
Yes.
Okay. Here is the new link in chat. So I'll work on getting this on the social media accounts so people are notified if you want to go ahead and get started.
Yeah. Well, hello, everyone. I am just going to ask everyone to turn their microphones off and their cameras off. And then when we start with the questions, you can turn them on again. We'll just wait a couple more minutes before we start with the cafe. Okay, so I will start. Welcome to AFO Café. This is our first cafe, and it's going to be a series of monthly events that we will be offering free and open to the public. And I am Valentina Ferretti. I'm the president of Association of Field Ornithologists. And for those of you who don't know AFO, I'll just give a brief introduction. We are a member-based organization, and we're focused on the study and conservation of birds and their natural habitats. And we view ourselves as the bridge between the professional and amateur ornithologists, and we have a strong focus also on supporting Latin American ornithologists through outreach, scientific sponsored memberships, and different grants. If you become, sorry, I keep this getting these screens opened up. If you become a member of AFO, with your membership, you support AFO grant programs for students and amateurs. You support avian conservation programs and also the Ornithological Council. And I think some of you might not know what the Ornithological Council is. So briefly, this is a council that's formed by member ornithological organizations that acts as a link between the scientific community and private and public decision makers. And it also provides support to ornithologists by informing and helping out with permits and animal welfare protocols. You also support with your membership the organization of our annual conferences and student and early professional events that we offer usually at our annual conference, and also travel and registration awards for the conferences. You also support diversity, equity, inclusion, and justice initiatives and the publication of the Journal of Field Ornithology. Now, members receive a discount on banding and field supplies from Avanet Research Supplies. They receive access to our Journal of Field Ornithology, access to our grant programs, discounts on conference registration, and also assistance with permits and animal care protocols through the Ornithological Council. So Avanet is our sponsor for the cafes, and I'm going to let Patrick Keenan from Avanet give a brief introduction of what they do. Patrick.
Thanks so much, Vale. I'm very happy to be here, and I think this is such an exciting step for AFO. For those of you who are not familiar with Avanet, Avanet is AFO's research supply arm. We support researchers around the world with mist nets and accessories, tools for banding and measurement, That is our specialty. We continue to broaden our product line to support field biologists in whatever capacity they need, including recently becoming a dealer of Vortex binoculars and other products. As a team, we're very passionate about research and conservation efforts, and we take a lot of pride in all of the work that you all do. This is a fantastic evening event, or it's evening where I am. And I encourage anyone to visit us at avanet.com, offer suggestions about products or solutions they found in the field. You can join us or engage with us on social media through Facebook or Instagram at Avanet Research Supplies. And we love also hearing about the successes and even some of the failures of research projects as well. So with that, I can't wait to hear more of this evening's speakers discussing something close to my heart personally, which is species delineation. So thank you so much, Vale.
Thank you, Patrick. Thanks. So with that, I'm going to introduce today's cafe. The title is going to be on the origins of a new species of seedeater from the Esteros del Iberá in Argentina. And Leonardo Campagna is going to present their work, and Cecilia Kopuchian, Adrián Di Giácomo, and also Melanie Browne are going to answer questions afterwards. So let me introduce the speakers first, and then I'll give you a brief instructions on how the cafes are going to work. Leonardo received his PhD from the University of Buenos Aires, Argentina, and then he did a postdoc at Queen's University and is now a research associate at the Fuller Evolutionary Biology program at the Cornell Lab of Ornithology. And Cecilia Kopuchian and Adrián Di Giácomo both are received their PhDs from the University of Buenos Aires in Argentina, and are now researchers at CONICET. This is in Argentina, the National Research Council in Argentina, and they are based in Corrientes, and at the institute that's CECOAL, it's Centro de Ecología Aplicada del Litoral. So let me say or ask again to please turn your microphones off and your cameras off. And Leonardo will talk for about 30 minutes and present their work. And after that, we'll open up for questions and a conversation. We are also live streaming this on YouTube. And so Rebecca Brasso, she's going to be reading the questions from the YouTube channel. So if you're on YouTube, please write your questions there, because we are also going to be able to answer some of your questions. And just as a reminder, this is an informal event, and we would like everyone to be able to participate and feel comfortable asking questions and talking afterwards. So I'm going to stop sharing my screen, and Leo, you can go ahead.
All right. Can you guys see that?
Yes.
And I'm not muted, right? Yeah. Let me just arrange the windows. There we go. Well, thank you, Ale. Thank you for the introduction, and thank you to everybody for inviting us to speak today. So instead of trying to do a talk where all 3 or 4 of us would speak, we voted, and you got stuck with the only person in this group that's really not a field ornithologist. So my main area of expertise is in genomics, but I will try to convey the general message of our talk. And of course, then we'll have time for questions. We can all answer questions. I also saw in the chat, people were asking whether the talk would be in Spanish or in English, and we're going to go with the talk in English, but we could also answer and translate questions in Spanish if people are more comfortable with that. So what I want to talk, before jumping into the main theme of today's talk about the Ibera seed eater, I want to talk a little bit about the radiation of cappuccinos, which are shown right here in this picture. And I've been studying them since my PhD. I've been interested in this group because it constitutes what we call a biological radiation. And this means a group in which speciation rates, so the rate at which species have been generated in time, is unusually high. And the reason we study this is because it's an exception, and we are trying to get an insight into the mechanisms, the genetic or ecological mechanisms that generate species, and try to see if we can learn something about the speciation process in the mix. Of course, the center of the talk is this species here, the Ibera seed eater, which was described by Adrián and Cecilia in the year 2016 in a paper, but they had observed it earlier on. And before I jump into the talk, I also want to acknowledge all the co-authors of this particular more recent paper that we published on this species, and especially Sheila Turbeck, who's now postdoctoral associate at Colorado State University, who on her bike in the Esteros del Iberá did a lot of the heavy lifting for the fieldwork involved in this project. Okay, so to jump in, I wanted to start by showing you a picture of a weird bird. And this is an ornithological crowd, so I'm sure you guys, or most of you know what this is. It's called an oil bird. And it's called an oil bird because, as many things, it was named after the function that humans initially found for it, which is they would harvest the juveniles that have a very large fatty reserve and use that for oil to burn in lamps. And this is a very strange bird. It's a cave nesting bird. It forages at night. And one of the strangest things it does, it can echolocate. So very much like a bat, it can emit high frequency sounds and listen for the echo and use that to forage at night. It's a very unusual trait in birds. This bird is maybe a little bit more well-known because it looks pretty weird. It's the hoatzin from the Amazon. I forgot to say that oil bird is also from northern South America. The hoatzin is a strange bird because it eats leaves and it ferments them. Very similar to the way that a cow ferments its food. So it sort of saves it in a crop in its foregut. And it's also called the stink bird, or known as the stink bird, because of course that emits a smell. And again, so fermenting leaves, that is something strange that not a lot of birds do. And so I want to segue into looking at this phylogeny, which is the phylogeny of all birds. And so this is a phylogenetic tree. If you're used to seeing these, you may notice particularly this one's, to fit it in the page, they made it into a circle. And so this depicts the relationships between all extant birds. And this tree has another layer of information on it. It has colors, and it's color coded in a heat map type way. So colder colors, blue, shows areas of the tree where the speciation rate is very low. So there are very few species generated in long periods of time. In contrast, you'd imagine the opposite. These warm colors show very large number of species that generate in a very short period of time. So that's the biological radiation I was alluding to before. And perhaps the oil bird and the hoatzin are such strange birds because they actually live on the two longest, most solitary branches of the bird tree of life. So they have no living relatives, and they have diverged from their closest relatives a very, very, very long time ago. So as I pointed out before, you can also see these areas of rapid radiations, biological radiations, such as Darwin's finches right here, this little red group here, or the white eyes that are known for being very, very speciose. And a lot of what I do is try to understand what are the processes behind these differences. So why do these speciation rate vary? Why are there long blue branches and very short red branches on this tree? And what are the processes that promote speciation? And I do it through the lens of this particular radiation that falls here. It's not well represented in this tree in particular, but it's the cappuccino seed eaters from South America. So just a little bit of context before we go forward. speciation usually proceeds, the vast majority of species are formed in a way called allopatric speciation. And this means that if we imagine we have two fish right here, there are two blue fish, and in the middle there's a mountain or some geographic barrier. It could be a river. There are many different geographic barriers that have contributed to it, glaciations, things like that. Over time, these populations are separated. And they're separated for so long that random mutations start to accumulate. And eventually, allopatric speciation is a waiting game. You separate two populations, you leave them in isolation for a very long time. And what most likely is going to happen is over that time, you'll have mutations and these things will diverge. And so eventually you have a blue fish and then you have an orange fish. And if this has gone on for long enough, if by any chance they come back into contact, when this happens, they no longer recognize each other. So allopatric speciation tends to be the norm, and it involves isolation and time. However, this brings us back to those rapid biological radiations and says, okay, what's different in this case? What process could be making this different? Are things isolated in space in many different places? Perhaps, but why have they accumulated so many differences so fast? So what I'll present today is this idea of a different type of model that could lead to speciation. And it's a model it has different names, but basically it involves reshuffling the genetic variation that you have to generate a combination of new things. So in this case, you have a blue fish, you have a yellow fish. They hybridize and generate all these different possibilities. Fishes that have a little yellow fin or a blue fin or things like that. And what will likely happen is most of these things will not go forward. It's sort of an evolutionary dead end. But eventually you may have processes like sexual selection that involve females choosing a certain appearance, a certain phenotype, and one of these will take off. And so it will let you generate many species in a very short period of time. So these are males from 10 different species of southern capuchinos. And you can see that they vary in different plumage patches, right? So you have the different color of a belly, different color of a throat. And so these things in a way are reshuffling, at least in the way they look, but then now we know in their genes too. There are mixing and matching of plumage patches from different species. And so what we think is going on in this radiation is that there has been mixing of the different gene pools over time through hybridization that have generated new color patterns. So different ways in which birds look, which eventually with song differences that are also another layer of complexity here, are enough to maintain these birds as separate species, or enough, at least at the beginning, to allow same to mate with the same. We call this prezygotic isolation. And so the idea is that this will provide a certain strength of isolation, and over time, additional mutations are gonna accumulate in these different lineages, and they will become increasingly different. So here are pictures of the 10 different species, and as I mentioned before, you can see how the different color patches vary. These are all adult males in reproductive plumage, and I should say that each one of these males has a specific song, or at least there is some geographic variation, but there are specific songs that also match up with the color pattern. So these are two aspects that differentiate the adult males. There actually is more coloration diversity within this radiation, and so there are many cases of alternative morphs that are usually rare or always rare, and sing the same color of sing the same song of the species they belong to. But this is just to show you that there is more variation in coloration that we think is not representative of distinct species. And this will be important because we are going to talk about a new species today. So in contrast to males, these species are sexually dimorphic, which means that females look different from males. And so the males are colorful in this case, and the females tend to have this brown or olive-like plumage. And they're actually, we cannot identify them easily through morphology. Also, juveniles look similar too. So it's actually very difficult to identify anything that's not an adult male. A few things about the general biology of these species. So they're what we call sympatric, meaning that their ranges tend to overlap. And so this is a heat map where I'm overlaying the ranges of all these different species. And you can see that they tend to all overlap. In particular here, we're going to get back to this area here, where you can find up to 7 different species in this area. One thing to note here is that even though they are sympatric right now, meaning that you can find them in the same place, this doesn't necessarily mean that they speciated in a sympatric way. There could have been events of past isolation that have contributed to this. What we do know is that these birds are sympatric and are very early in their divergence. And so we know that the way we do this is we use molecular sequences that mutate at a certain rate. And so we compare those from within the capuchinos to their closest relative and see how different they are. And so we sort of back calculate and imagine, okay, if this mutates at this rate and it's this different, therefore they must have been isolated for this amount of time. So that's the rationale for that analysis. And if we do that, we can date the radiation to somewhere in the Pleistocene. There's caveats here, but we know that most of this diversity must have generated within the last 1,000,000 years, which in evolutionary time is not that much. There are variations of this that make us think that they may actually be a lot more recent, but this is a pretty conservative estimate. So I mentioned they're sympatric, and you could have a situation in which they inhabit the same forest, but you have understory birds and then canopy birds. So in a sense, they're found in the same place, but not at a microhabitat level. These birds are sympatric in grasslands, and so this is a structurally relatively simple habitat, and we do find them breeding in the same areas, in areas like this of the grasslands where there are no barriers, or at least obvious barriers. There is a discussion of how, if there is some type of ecological specialization within the group, it could be true. We don't yet have the data to necessarily show that. So my point here is they inhabit a relatively simple habitat, and they breed very closely together or side by side in some cases. Oops, there we go. A few more things about their biology. So they show very little morphological variation. So if you measure their beaks or different aspects of their beaks, they're not like Darwin's finch radiation where you have beaks of different sizes, different depths, and things like that. They consume the same foods, so morphologically they're very similar. I mentioned before that the males have different songs, and in this type of songbird, this is a learned trait. So this is actually, there are debates of what proportion, or if there's a genetic component to learning, and there clearly must be. But as far as we know, they learn their songs. And I didn't want to try to broadcast their songs and make it work well on the Zoom feed. I didn't know if I could do it from my computer. But Vale, if you can just drop in the chat 2 links, just in case you guys want to explore songs from different birds, you can just follow those links to the Macaulay library. One more point. As far as we know, they can generate viable hybrids. So from data from aviaries, we know that you can cross these birds and they can generate a viable hybrid F1. That bird seems to survive as well as the pure parentals. So, and this probably happens in the field too. It's a little harder to identify them and to be sure if that is fully mature plumage. But this clearly happens in the field too. And this is something that we expect with recent species. So we expect them to be able to still hybridize, do that mix and match of things. And then it may be that those hybrids actually don't do that well because they have intermediate plumages or the mismatched song or things like that. So the next couple of slides are perhaps a little technical, but I'll walk you through them. And the first thing I wanted to show is how we realize that this is an important problem, or how we know that this is a radiation where species generated very fast. And so we did this by trying to generate a phylogenetic tree. And if you haven't seen these before, a phylogenetic tree basically just shows you how species are related to each other. So if you follow here, which is what we expect phylogenetic trees to look like, let's look at this cartoon. You usually expect to have, a species that split off from everything else, and then in a very arranged way, you can see how species affinities, right? There are cases in which you cannot resolve the relationship between species, and we call those polytomies. And so here is a polytomy within a tree that's partially resolved, as the example says. So, we can see things branch off, and then all of a sudden we reach this area that's called a node, where everything sort of is equally related to each other. An extreme example of this is what we call a star tree, where basically there are no relationships here. Everybody is equally related to each other. Now, if we build a tree for the capuchinos, and we did that using many different genomic techniques and sort of over basically a period of about a decade as the genetic techniques changed, our result was always the same one. And it's, if you look up here, these little lines represent different individuals of the same species that is labeled. So you can see that this part of the tree is resolved as we go on, and we resolve it up to this species. And then all of a sudden, everything here, so the 10 species that are found here, are all mixed up. So it's a situation, basically a polytomy within the tree. And so when this happens, there are a few processes can make this happen, but one of them is that you're actually very early in speciation. And so there are very, very few differences between the species. And as I told you a second ago, we sort of like grew in the resolution as things moved forward, but we were never able to basically find a genetic difference between species. Until about in 2015, we for the first time were able to sequence entire genomes. So this is all the genetic material in the bird's genome. And so for the first time, we were able to take genetic data and ask this analysis, okay, here are 70 individuals, and the analysis doesn't know what species they belong to. As you imagine, the individuals are the circles and the species identity are the colors. And you tell this analysis, see if you can cluster out individuals and let's see if they match their species identity. So we did that. And for the first time, we see that, for example, here, all the Nigra rufa individuals grouped together. And the same thing happened for the other 5 species. And so what this is telling us is that in the entire genomes, there is information of species differences. We had never been able to find them, and which this probably means that there are very few and far between. And so as an evolution biologist, I'm interested in finding those areas and trying to see what genes are in those very narrow areas, because the rationale behind that is that if we know which genes are different early in speciation, well, perhaps we can make a conclusion about the importance of those types of genes to speciation. Are these really speciation genes that can, when they change, they fuel speciation and we get more species? So here, these are the last couple of more technical slides that I'll show. But what we do to do this, the technique, I sort of have to explain it so it makes sense, is we can take all the genes in the genome and sort of line them up horizontally. And we do that between 2 groups, right? So here on this axis, there are all the genes as we go on. And then we have a measure of how similar down here to 0 or how different up at 1 they are. And so we take species A, species B, many individuals of each, we compare them. And as we move on, we say, oh, this is probably the same, almost the same, almost the same. Here you hit a little difference. You guys see that peak. And then eventually in this cartoon, you have 2 large peaks. these are the areas of the genome that are different between these species. This is where the information is coming so that we can cluster them apart. And this must house genes that are important to making species different. You know, the alternative is you come back in a situation where you compare two things that are already too different, two species that are well established, have been diverged for a very long time. And if you do that, when you move down the genome, you can see how everything's like higher and a little bit noisier. It's very difficult to tie a situation like this back to the original genes that made these two things different. So we want to focus on a case like the one on the top, and the cappuccinos seem to be that. So I'm going to make this graph with the data that we generated, the whole genome data that we generated, and this is how it looks. Basically, I can take the two species that are the most different to each other, and they're different in only 0.3% of their genome. The two more similar species are different in 10 times less, so 0.03%. Very narrow areas that are depicted here as going over this certain threshold. And in total, across all the species that we compared, we only found 25 different areas of the genome that make them different. And so my work in genomics basically is trying to understand the processes that generate these different areas of the genome. But the more important thing for here is we want to ask, as I mentioned before, what genes are under these areas that they're called divergence peaks. And so if we look across those 25 different areas, there are a few hundred genes in total, but the most represented, there's about 10 genes that belong to this pathway here. And most of you may not be familiar with looking at biochemical pathways, but they basically show how different molecules interact to make a certain product. This biochemical pathway in particular is called the melanogenesis pathway. It's what produces melanins, eumelanin, pheomelanin. So they were talking about black colors, yellowish, reddish colors. And this pathway is quite conserved. It's sort of the same general pathway, makes colors in fishes and humans, in many, many birds. And so the most represented pathway, or in the differences among these cappuccinos, is this melanogenesis pathway, with many, many different parts of it are different. So they're sort of fine-tuned in the different species to produce these different colors. And this obviously acts when the feathers are generating and depositing pigment. And this is not, as I just mentioned, is not unique to birds. These same genes are involved in differences in many domestic animals and wild animals, at the species level, within the species, or even higher up. So it seems to be, coloration seems to be important across many different organisms, but also particularly among recently divergent birds. So these are all examples of recent species of birds that have very, very little differences across their genome, but they are mostly or only differentiated at coloration genes, just like the cappuccinos. Okay, so let's move on to the main part of the talk now that I've given you a little bit of background about, in a way, the things we needed to know so that when Adrián and Cecilia came across this new color pattern, we sort of already had a hypothesis about how this could have been generated. So the story continues here in the Esteros del Iberá marshes in northeastern Argentina. Right here, this is a big wetland. And here you can see sort of a zoom in, the Paraná River. And throughout its geological history, this river has changed its course many times. And just by looking at it, you can see that it's left its mark on the landscape, right? These are all generally flooded areas like this, and then you have more extensive grasslands. Many of you maybe don't know that this is a very large national park now in Argentina. And I had pointed before to just pay attention to this area where you see the highest accumulation of cappuccino species is actually coincident with this part. So it's right here, and it's showing us that this is probably the epicenter of diversity for this group. And so has this nice logo with is it called the strange-tailed tyrant, I think in English. And this is fitting because about in 2001, Adrián was starting his PhD working exactly on that species and other grassland birds. And he encountered this bird here. So this bird is now the Ibera seedeater, and as you can see, it has certain components of other species. So, you know, it has a white belly, sort of similar to the pearly-bellied seedeater here, but it has a grayish cap, so it doesn't match that there. The dark-throated seedeater does have a grayish cap and also has the black throat. So as I've been mentioning before, it's a little bit of a remix of other species, so a mashup of things, of traits that we see that we already knew in other species. And so when I first heard about this, the first thing I thought about were those morphs, sort of low frequency morphs that I showed you at the beginning of the talk. So, you know, within species, you have some degree of color variation. However, Adrián was quick to point out, it's like, well, there are some key differences. And first of all, this bird has its own song, which those morphs don't have because they sing the main song of their species. And the second point is, well, a lot of those morphs are rare and are found at very low frequencies. However, where this bird occurs, and it doesn't occur in many areas, it is actually found at very high densities. So these reasons were enough. Oh, before I go further, this is actually a picture from Adrián's notebook from the first time he encountered this species in the field. And I think for you field ornithologists, I think it stresses the importance of art and being a good artist. Maybe you guys are a little bit better than this, Adrián. I'm not picking on you, but anyway, just kidding. But this is actually a piece of Iberaensis history right here. So based on what I told you before, Adrián and his collaborator and partner in life, Cecilia, in 2016 published a description of the species based on their differentiation in color, their high abundance, and the differentiation in song. And now, before I continue, so based on this, we were really interested in trying to understand genetically how the species came to be. And so first I want to show you some eBird data here. You guys are probably familiar with eBird, but these data, this graph right here is based on observation. So on checklists that have been submitted, and we waited, and you know, since this bird was first reported or people started to look for it in the early 2000s, we waited a good 10 years. eBird Argentina started, of course, not a little earlier than this. But so we gave some time for the database to kind of stabilize, and we're just plotting the probability of reporting. So if you'd like, it's probably a measure of abundance, or but it's measured as the probability of reporting a certain species in this area, the area around Ibera, for all cappuccinos. So you can see that basically there is a very common cappuccino, which is Sporophylla boxanta. And then all others are not as common, so they're found, the probability of reporting is actually lower. And you can see in blue is the Ibera seedeater, how it went from being the least common seedeater to now growing in abundance, and it's no longer the rarest one in the area. And so just to zoom in, if we take out E. boxanta, we can really see the scale change, and you can see the significant increase in about the last 5 years. So I've shown you where this place is, I've shown you the habitat, and this is just a close-up of the nests that Sheila and Melanie and others found while doing their fieldwork. From now on, I'll code the Ibera seedeater in blue, so Sporophylla ibenensis in blue, and in red, Spraflipoxantha. And so you can see that these things are really at a scale of 500 meters. They're really breeding close to each other. And they're not only in the same area, but these histograms are showing over 2 field seasons when the nests were found. Again, the same color coding, and the purple is overlap. So you can see that they're breeding at the same time. Just a quick look into the genomics. We compare these two things and we find again these 3 areas of the genome that make them different. Don't pay a lot of attention to this. These were areas that we know differed among cappuccinos in general and contain coloration genes from that melanogenesis pathway. So the same kind of thing. They're mostly differentiated in the areas that we know are different in the radiation. And so let me see time. I'll move a little faster for this. The most basic question is, okay, they breed at the same place at the same time. You know, do Iberaensis males and females mate together to the exclusion of the Ipoxantha males and females that are right by each other? Or do we have a situation where everything's mixing? So we call this assortative mating. So does the same mate with the same? And you'd usually be able to do that by going out and looking at the female of one species and the male and seeing if they're pairing together. However, as I mentioned before, the females have the same coloration and we can't identify them. And this is just showing a spectrophotometric analysis. So showing you across all these wavelengths how these species differ. And you can see the take home is that all these curves are overlaid on top of each other. So we can't really distinguish the females. So how do we identify females to know if the females of Iberaensis are breeding with the males of Iberaensis that have a specific and diagnostic color? The way we did that is by looking at these few areas that were different in the genome. And so you don't have to pay attention to the details of these plots, but it basically shows you males and females that were paired together. So in Iberaensis, the dark blue and the light blue tend to cluster together to the exclusion of the red and orange, which are Ipoxantha males and females. And we can do this over a larger sample. This was done with entire genomes. This is done with a sampling of the genome that actually was sufficient to separate things. And we're also including nestlings in here. So basically, if we use the genes, we can realize that even though they breed in the same place and at the same time, they can distinguish each other and choose who to mate with. And they choose their own species. Just another methodological hurdle is, it turns out that of course, you guys know that most of the males in a lot of birds, your genetic mate and your social mate is not the same. So there's a lot of extra pair paternity. In this bird, in these species, we calculated about over 50%. And so it's not enough to know which male and female are paired together. You actually need to know which male fertilized the eggs of that female. In a lot of cases, it's a male that wasn't paired with her. Well, because the sampling was good enough to actually look at many males, we could assign paternity to the eggs and realize that when it was a male from that was not in the couple, it was always from the same species. So this assortative mating does not break down through extra pair mates. And so let's see. Yeah, maybe this is the last thing I'll show you, the last piece of data at least that I'll show you. Well, we wanted to know if these coloration and song differences were sufficient for birds to distinguish each other. And the way we did that is through playback experiments in the field. So Sheila went out and she put out these 3D printed mounts that she painted. And at the same time, she was playing song. And we tried an Iberaensis mount with an Iberaensis song, an Ipoxantha mount with an Ipoxantha song, and a control of a closely but a little bit more distantly related species that we thought the birds shouldn't really care about, right? And so the idea of this experiment is to try to get a male mad. And if a male, the more pissed off that male gets, the inference is it's because there's an intruder in its territory and it's a sexual competitor and it wants to kick it out. If the male doesn't care, we're like, well, it knows that that is not its species, right? So we're gonna set up an experiment like that. And here are just some shots to show you, you'd imagine the birds would realize that this is a mount that has been painted. But as you can see, they get really, really annoyed at it. And so here, these two graphs that I'll show you is first, this is the focal species, so the responder, and we are exposing it to different treatments. We're exposing it to conspecific song and plumage together, or on the other extreme, heterospecific. In this case, to Ecosanta, we're going to expose it to Iberaenses. We're going to mismatch the treatments here, and then we'll have the control, which we know they shouldn't respond to. And we're going to do the same for the other species, right? But you flip it. In this case, the conspecific is Iberaenses. And so we're predicting that the strongest response is going to be to their own, and that's going to mean that they really care about other males invading their territory, but they will not care about the other species because they can distinguish it through color and song. So if we do that, that's exactly what we observe. The strongest, I forgot to say that this axis is just a measure of how mad they're getting or how much they're responding. Strongest response to conspecific, you mismatch, it's a little lower, significantly lower, and then you cannot distinguish the control from the heterospecific Capuchina. Same in the other species. So we know that song and color are important for species discrimination, at least through the behavior of males. We infer that perhaps the females too, and that's why we see assortative mating. I'm going to skip these because I think it will be more interesting to have time for questions. I'll skip these two and I'll just wrap up. The idea here, again, that I want to mention is that what probably happened here is that we had hybridization at some point and the mixing of a few different gene pools that resulted in a novel bird. You know, maybe this was generated. And then eventually we may have had a period of isolation. It may have been reinforced through song differences. There was selection, probably through female choice, and this became a new species. 3 points to close. The first one is, I think this illustrates a general process, and it tells us how these differences in color and also in song, which actually could be cultural, are enough for species to become isolated reproductively. And this can kick off the speciation process. We don't know where this is going to go in 10,000 years and 100,000 years, but we think it illustrates a mechanism of how you can generate species. The second point is that hybridization probably is helping out here, and it may be more common than we used to think. I mean, we know it's more common. There are many cases of biological radiations that have been actually fueled through hybridization. We have cichlids, heliconius butterflies, many different examples in birds. And the final question, I think, which I think about a lot, is will these incipient cappuccino species last? You know, aside from conservation issues, which of course are a huge issue, but would they last on their own? And I really like this paper for those that are interested in this thing, in these type of things, that it sort of asks, when we estimate these speciation rates from different sources of evidence, mathematical models, trees, the fossil record, we get wildly different estimates. And why could that be? And these authors propose that actually, they call it the ephemeral speciation model. And what they're saying is, well, actually, probably speciation is very common in nature, but the species that are produced almost never persist long enough. And I think this general process is showing you how you can get this process started, this general mechanism. But whether they persist or not will depend on many different factors. And that's something to think about. And so, okay, so we have time for questions. Just want to thank all my co-authors from previous studies, funding sources, and some of the images I used from illustrators, and also the drone video. And let's jump to questions so everybody can get a chance here.
Thank you. Thanks, Leo. This was a wonderful talk. While we wait for some people to write their questions, I just want to say, actually not write the questions. If you have a question, please raise your hands. You can use the Zoom feature to raise your hand, and we will be calling you to ask your questions. I think Cecilia and Adrián, well, and Leo too, they can all answer questions in Spanish too, but we will try to translate. And I just wanted to ask shortly Cecilia and Adrián if they can briefly just comment on their paper, their description of the species in their paper. And maybe they want to do it in Spanish and we can translate. Don't know if they... Ceci and Adrián, did you hear me? Okay, there.
We lost
Ahí
them.
estamos. Bueno, simplemente contar que fue una experiencia que se fue dando progresivamente, como explicó Leo, con observaciones iniciales que hace cualquiera en el campo. Muchos habíamos observado estos individuos, pero los asociábamos con plumajes de juveniles de otras especies, o mudando, ejemplares que están mudando. Y como comentaba Leo, la primera pista de que se trataba de que no eran juveniles era volver a los mismos sitios de muestreo cada año y encontrar individuos cantando con plumaje siempre igual y como los adultos que conocemos hoy. Esa es la síntesis de cómo fuimos empezando este trabajo.
Gracias. I think we have a question. Rebecca, do you want to read a question from the YouTube channel?
I do. So, question from Dan Baldessari on YouTube asks, what do we know about their historical biogeography? Is this true sympatric speciation?
I can jump in, and maybe if you guys want to also comment on it. I think this is a question that we tend to get. You know, I think what we can say, given our data, is that these are very recent species and that they are now highly sympatric, dependent on the comparison that you make. You know, making the inference that they were equally sympatric 10,000 years ago is speculative, so we don't know. We know that this area has a very dynamic geological history, and there are various processes that could have stimulated different levels of isolation. So I would be, you know, I think I want to be agnostic about whether I call this sympatric speciation or not. I think one way or the other, our data don't necessarily let us say that, other than they are recent right now and they've come back into sympatry at an early stage of speciation.
Awesome. Well, Dan, if you have a follow-up, feel free to type it into YouTube and we'll follow up with you. Maria Rodriguez, if you want to unmute yourself, there seems like you have a question.
Yeah, Maria Paula Rodriguez.
Yes, hello, do you hear me?
Yes.
Okay, first of all, Leonardo, thank you for your amazing talk. I really like this group of birds. So I have one question, is that first, well, it is a cool genomic work to find the genetic basis of the differentiation, no? Related to the coloration. But given that all males of the different species look the same and that the juvenile males look like the females, maybe there are differences that are related with genetic expression. So I was wondering if you plan to do like transcriptomic work to find which are those differences in the coloration between sexes.
Yeah, absolutely. So that's a great question. I didn't get into, you know, these areas of the genome that are diverged, but 99% of them fall in what we call non-coding regions. So they don't fall on genes, they fall close to genes. And we know from genomics and, you know, molecular biology that there are areas close to genes that regulate the expression of genes. So this is another layer on top of it. They're not different in the genes themselves. They're different in a coding region, or sorry, in a non-coding region that may control genetic expression. And so yes, the follow-up from a genomic point of view, there's a few different things that can be done. Transcriptomics is what I'm trying to focus on right now. And then there's also many methods of developmental biology that allow you to assess that type of stuff. So yeah, it's another layer that allows rapid speciation, where it's not to make the genes themselves different, but to make those shorter sequences that control the expression of those conserved genes different. And so that's it. Yeah, I didn't get into that, but that's a cool question. Thanks.
Okay,
Maria Paula, oh, I was going to ask if you had a follow-up, or that's it?
No, it was it, but maybe I think that it would be cool to compare between sexes, no, and between the different, the juveniles and the adults. Or I think that.
Yeah, gene expression differences, you know, you can compare males to females. You can compare male reproductive molt to male post-reproductive molt. And it allows you to do very different comparisons that let you sort of assess which genes are changing their expression to lead to the coloration differences. Yeah.
Rebecca, do you have another question from the YouTube channel?
I do not. I do not. So again, if we want to open it up to, you know, all of our guests here on Zoom, please feel free to raise your hand or speak up with a question.
Yeah, I'm going to read Diego Gallego's question. He asked, so you can say that Iberaensis may not survive as species? Leo, you answered it, but maybe we can answer it to so that everyone will.
I have my version of that, but then I'll pass it on to Adrián, that I'm sure he's gonna. What I'm trying to say here is that reproductive isolation is not complete until, you know, you have what's called post-zygotic reproductive isolation, where you actually hybridize and things are not compatible because the way in which their genes work is just so different that they can't come back together. And so until that happens, which is usually a very large period of time because it involves random mutations in random places of the genome, you usually say the speciation process is ongoing. And that's why I think here we're in a point in which we're showing sort of, this is how it starts. Where it ends, we don't know. What I mentioned in the chat is I wouldn't put Iberaensis at a different level of any of the other species. And now what I wanted to pass it on to Adrián is like, say you said 100,000 years. Adrián, do we have 100,000 years of grasslands at the rhythm that we're going?
Es más probable que la especie desaparezca por el cambio climático que por un proceso evolutivo. Publicamos con Melanie hace unas semanas un artículo sobre la biología reproductiva de esta especie, y uno de los factores que afecta a la reproducción de esta especie es probablemente la existencia de agua en los pastizales a donde se reproduce. En temporadas extremadamente secas, estas aves frenan su reproducción, es decir, que pueden no tener una buena producción de pichones para la próxima generación. Y bueno, y este tipo de fenómenos podrían de alguna manera afectar rápidamente a una población tan pequeña.
I'm going to translate that. I didn't translate your first answer, but so it's, according to Adrián, it's more likely that they're going to be disappearing, not because of evolutionary processes in the species, but more likely because of climate change, because they're highly dependent on water for their reproduction. And in years of drought, they don't reproduce, or their reproductive success is quite low. That's good. Okay. Any other, Diego, do you have a follow-up or no? So I have a question. There's hybridization, or at least you propose that there's hybridization and now there's strong assortative mating. Do we expect this strong assortative mating to relax at some point or not? Or what would happen in the future? Or what can we predict to happen?
I mean, I think we've observed over 2 years, we didn't find a hybrid couple, but that's a pretty short period of time. If we actually look at the structure of some of these regions that are most divergent, we can see that we have generally epoxantha versions of these haplotypes, so these long portions of DNA, and Iberaensis versions. And then you have, in a smaller proportion, the mixed up recombined versions. And so that tells you that, these things in the past have hybridized at some point. We know it's perhaps low enough that we didn't detect it in the years that fieldwork was done. But I think you probably, even with pretty strong reproductive isolation, you may have, it only takes, one hybridization event for the gene pools to start mixing and matching. And, we have a snapshot of what things looked now, but we don't know, how it went in other localities and over long periods of time. There's much to still know about that.
Thank you. Rebecca, do you have a question?
Sure, I do. I have a question from Manuel Sanchez from YouTube. So Manuel asks, what about food availability, distinctive diet preferences, and nesting area preferences? Can it cause plumage variation in carotenoids and speciation?
Adrián, do you want to talk about nesting and diet, and I'll talk about plumage later.
Se sabe que los capuchinos en general se alimentan de semillas de pastos nativos y son muy dependientes de estas semillas durante el periodo reproductivo. Casi con seguridad, donde hay semillas van a establecer sus territorios, pero al menos lo que vemos en el mapeo de nidos y de territorios es que están en los mismos pastizales de similar estructura y la alimentación es un tema pendiente para estudiar. Hemos hablado hace unas semanas con Leo de este tema, y porque puede seguramente deparar algunas nuevas preguntas acerca de tanto de la ecología o de la evolución de este grupo. Es necesario hacer un estudio comparativo de las dietas y me parece, tal como hicieron la pregunta, muy interesante también evaluar el tema de los aspectos nutricionales que pueden influir en estas especies que tienen plumajes tan característicos, pigmentados.
Let me translate, Leo, before
you
answer. So Adrián said that these birds feed on native grasslands, and they're very dependent on the seeds of these native grassland species for their reproduction. But they breed in grasslands that are similar in their structure. Is that right? I think. The different species breed in grasslands that are similar in their structure. So what's pending is to study the food and maybe work on a comparative study of their diets and also the nutritional aspects of those, of what they feed on.
And what I wanted to sort of add on is the second part of the question, can, for example, diet cause plumage variation? And so this comes from carotenoid coloration, where a lot of the direct molecule that changes color, like think of flamingo, or a molecule that is then converted gives the coloration plumage of the bird. And so in this case, these are, we've actually analyzed chemically the feathers of all the different species. All the coloration is melanin based. So it's either pheomelanin or eumelanin, and it's endogenously produced. It doesn't come from the diet. And it's different ratios of those two kinds of melanins that dictate the color. And so what I was thinking is interesting about the question is that there are also properties of the melanic coloration of the feathers that are generally associated with different type of habitats. So you have more melanized feathers tend to be found in wetter environments. And it's thought because that gives more resistance, and it's more resistant to bacterial degradation and other things. And that's a pattern that's known in birds. I had never really thought about that in this context. If you would have a bird that's sort of ecologically more inclined to being in wetter areas, if they are in fact darker. I don't think we probably have the sort of the distribution of species in habitats to test that necessarily. And I'll just mention there's another question in the chat that essentially is, do recognized species exhibit ecological differentiation? And that's what I think, and Adrián was alluding to this, we're lacking, I think, through observational studies and a few more preliminary studies that are out there. There could be, I wouldn't rule it out, but we're still lacking sort of a comprehensive systematic study that allows us to assess sort of smaller scale ecological differentiation.
Thank you. There's another question from YouTube from Justin Mann. What behaviors were included in the response measure, PCA, during the playback experiment, and were they scored based on aggressiveness?
Yeah, so we took, Sheila did these experiments. She took various measures of aggressive response. So, number of flights, proportion of time spent close to the mount, number of flights over the mount, whether it would directly attack it or not. And I think she has, and we had a previous study too, where we had done it with other species, where we take basically measures of proximity or times it's spent attacking or directly attacking the mount. And then we take those variables and we sort of extract the principal component. So we crunch down the dimensionality of that. And that is our PC response. So it's basically, it gives you a higher score the more time the bird has attacked mounts or flown over it or spent time really close to the mount.
Thank you. Maria Paula, you have a question? You're raising your hand?
Yes, I have another question. I want to ask, related to the paternity test, in which stage of the offspring do you do the sequencing? If you collect eggs or hatchlings, fledglings?
I don't know if Melanie is still around, but probably she's the best one to answer that.
Hi, yes, I'm here.
Do you want to take that?
Sure, yes. So what Sheila did is she took blood samples of nestlings, actually. So when they were around 6 days old. And I believe most of the samples were taken from blood samples and some from unhatched eggs also. But those were 2, I think, or around that.
Okay, thank you. Is that,
yeah.
I was thinking that, well, you showed that there is a strong pre-mating isolation between these two species, no? But given that we cannot know a female, of which species she belongs to, maybe there are females that are of one species and they copulate with males of other species, but due to that reshuffling of their genomes, there could be also post-zygotic incompatibilities. So that would be cool to study also, no?
Yeah, so the only measure we could have of the post-zygotic incompatibilities was through an experiment that we did with captive birds. And it's actually quite difficult to do. We saw that the hatching success or things like that of F1 hybrids versus pures didn't differ. You'd expect actually, from theory to those incompatibilities to show more in the F2s, but it was actually, we didn't have the sample size to do that. But this is, that's a big question to answer in the future, I think.
Thank you. There's another question from Diego Gallegos. I can read it, Diego, if you want. It says, I have heard the hypothesis that the lost type of ruficollis could be an iberaensis as a morph of the typical ruficollis and be the only known individual of Sporophylla melanops, is a morph of iberaensis. What do you think?
When you say the lost type, you mean like the paratype, the type specimen in a museum when the species was described? If you mean that, then I don't know. I can't speak to that, and I also can't speak to melanops either. Yeah, to the original type. No, I don't know if those. What I can say is that iberaensis is different from. Those are the slides I actually skipped because we were running out of time. But we can show that in those three main areas of the genome that are different between Hipoxantha and iberaensis, we can show that in one of them, iberaensis shares the genetic variation with ruficollis, and in the other one, it's completely different from ruficollis. So it doesn't. We can, if you give me a DNA sample, I know how to, where to look to distinguish iberaensis from ruficollis from a genetic perspective. The history behind where these types, what they are, we could sequence them now and figure out, but I don't really have an opinion, a formed opinion on it.
Thank you. And I'm going to read Diego Oscar's question. This is our last question. Or if you have another question, we can answer one more. But Diego asked, Leo, the Ibera Seedeater that can be found in Corrientes are the same that are found in Paraguay? You know if this is migration? Do we know that?
Adrián, what do you think?
Leo, Adrián.
Sí, la especie está un poco más ampliamente distribuida a lo largo de su migración. Conocemos el área de reproducción en Corrientes. Hay algunos registros de nidificación o de adultos alimentando pichones en Paraguay. Y es la misma especie, son parte de la misma población. Todavía no se conoce el destino final de la migración. Hay algunos registros en Bolivia, en el Beni y en Brasil, pero por dónde es la migración no tenemos detalles.
See, and I would add that even, not only thinking about iberaensis, but thinking about all capuchinos in general, there's a lot of geographic variation within the same species. I think the person that's done the best sampling of that is Marcio Repenin and Carla Fontana, and they can show in different species, this is not published yet, but they can show that there is genetic variation within these more widespread species, so not necessarily iberaensis, but there's also a lot of variation in song. So, as you ask, is the one in Paraguay the same as the one in Ibera? And maybe those populations are connected, but I wouldn't expect there to be population connectivity necessarily at a larger scale in other species. Because as we know, they tend to be phylopatric. There are song differences, and those will sort of tell us, they foreshadow what the genes will tell us eventually, because cultural mutations and cultural drift is faster. But it's surely there's a lot of within species variation too.
There's Carla is in the audience too. So you mentioned Carla Fontana, so she's listening. I don't know if she has anything to add, but No. Okay. So if there are no more questions, are there any other questions, Rebecca?
There's one more question in the
Okay.
chat.
Just you can
It's in Spanish.
Oh, here?
Yes.
Well, it says from Jonas Rossoni.
Yes.
Hola, Leo. Congratulations for your work. I've been studying the reproductive biology of Cinnamomea here in southern Brazil, and I've been marking adult males. And I noted from a year to the next that some of them show the characteristic plumage of the species, all brown. And in the next year, they only have, and in the next year, they have the throat marked or with brown spots, dots. Is this some mechanism related to the diet, like carotenoids?
That's a cool observation. I mean, I don't think this has to do with carotenoids. I think because, as I mentioned before, it's a melanin-based coloration. I wonder, you know, this is speculative, but I know it happens, for example, in fairy wrens where the varying levels of testosterone affect the coloration of the birds across their lifetime, but also across years. I wonder if there's a sort of a condition-dependent sort of hormonal context that is affecting coloration. I don't know. That's very interesting to know. Maybe I'll write to you and ask more.
Okay, I think that was our last question. And well, thank you so much. This was an amazing talk, and thank you. This was our first cafe, and it was great having you all here to participate. So I'm trying to find the clapping thing here, but I can't, the reaction.
Thanks so much for the invitation.
There. And I just want to tell everyone that our next cafe is going to be on July 30 at 4 p.m. Eastern time. And we're going to have Dan Baldassare talk about northern cardinals. Does living in the city make northern cardinals change color? So I hope you sign up. We're going to post that on our social media soon. And I hope you join our cafes. And thank you very much. And thank you, Avanet, for being the sponsor to our cafes too. Thanks.